Good afternoon, everyone. Welcome and thank you for joining us for this new RedTalk webinar. So I am Aurore Touzart, I'm a medical doctor and biologist in the Onco Lab in Necker Hospital in Paris. So RedTalks is a webinar series dedicated to open innovation in lymphoid neoplasm organized by the Carnot-Kalim Institute. Our goal is to bring together experts from academia and industry to discuss the latest breakthrough and challenges in the field. So now I will let our Professor Romain Guièze to introduce our speaker, so Professor Iñaki Martín-Subero.
Good afternoon. I'm Romain Guièze, so hematologist in Clermont-Ferrand in France and co-chair of the CLL committee of the LISA group. It's my great pleasure to welcome Professor Iñaki Martín-Subero, who is internationally recognized for his major contributions to the field of epigenomics in lymphoid malignancies. So he leads a research group at the University of Barcelona as an ECREA research professor at the IDIBAPS of Barcelona. So in this episode, we will explore how cutting-edge epigenetic approaches can retrace the evolutionary history of cancer, opening new avenues with potential clinical application. So Iñaki, please, you can start your presentation.
Thank you so much to you both for the introduction. Thank you very much to the organizers for inviting me to this very nice series of talks. So my duty today is to explain to you a little bit some of the things we have been doing in our laboratory over the last years. So we do epigenetics in lymphoid tumors. When I started the lab, I had this very clear idea that epigenetics is the major gene regulation mechanism. And we started the lab trying to identify genes that become deregulated in lymphoid tumors, etc. But gradually the data started to tell us something different. We do in part hypothesis-driven research, but also in part data-driven research. So what I'm going to tell you today is something we could not anticipate before. It was not out of my imagination that this came into life. It was more that the data were telling us this story. And so the title of my talk is Blended Epigenetic Memory in lymphoid tumors, and which are the biological and clinical insights. And this requires a little bit of introduction. The first thing is that I will focus my talk on chronic lymphocytic leukemia. So brief introduction on this disease. It is a basal tumor. Many of you will know it's a quite frequent leukemia in the Western countries. And what it is perhaps more characteristic of the disease is that the clinical evolutionary paths are quite variable. So we have a fraction of patients in which the disease is diagnosed and there is no need for treatment, and the disease gradually and spontaneously, without any treatment, disappears. There's a very low fraction of cases, mostly of IGHV mutated, that regress spontaneously without treatment. There is a group of patients that remain stable for even a decade or two. There is some patients in which the disease can progress, requires treatment. Some respond well, others respond a bit worse. Eventually, those that respond well may relapse. And even there is a fraction of patients that is the other side of the spectrum of the clinical behavior of CLL. That is the Richter transformation. That is a very poor prognosis condition in the context of a CLL, which is the transformation of the CLL cells, which are small round cells, to larger cells to give rise to frequently diffuse large B-cell lymphoma-like disease. And in this context, when these cells transform, this is really a dismal prognosis and quite difficult to treat. So perhaps the most relevant biological feature that helps us to discriminate between good and bad prognosis in CLL is the mutational status of the immunoglobulins. This is what is used in the clinics together with some set of mutations. We have those CLLs whose neoplastic cells show somatic hypermutation of the IHP locus, and these ones have a better prognosis than those cells that don't have the somatic hypermutation, which is basically reflecting whether the cell of origin has experienced the germinal center reaction or not. The second aspect I would like to introduce is epigenetics, and basically epigenetics is classically defined as the science of gene regulation, but I would like to go a bit beyond that, and it's more the science of genome function, or how different parts of the genome can provide information that is manifested into a phenotype. And not only that, but the methylation is also, or DNA method, or let's say epigenetics is also a way to register what cells have experienced in the past. So the information is coded in 3 dimensions. So we have the DNA methylation, also the hydroxymethylation at the 1-dimensional level. In 2 dimensions, we can have like the histone modifications. Histones are not only important to pack the 2 meters of DNA that we have in each cell into the tiny cell nucleus, they also play a very important role into determining the degree of compactation of the chromatin. If it is very compacted, the message that is contained in DNA will not be read, and the genes will be silenced. Whereas if it is more loose, more open, then regulatory elements, transcription factors can bind, polymerases can start doing their job and to transcribe genes, and therefore the different histone modifications and the overall accessibility of the chromatin play an important role in regulating the gene expression. And then we have the 3-dimensional interactions, in which distant regulatory elements can affect gene expression through looping of the chromatin. So this is a list here of the most, let's say, of the canonical epigenetic marks, at least those used in the reference epigenome by the Human International Consortium for the epigenome. And we see, for instance, the genome is basically information storage, and then we need to give it function. So how do we give function? Well, through the integration of multiple epigenetic layers. So we have DNA methylation that has traditionally been related to gene repression. However, when I write in the function here, context dependent, is that, if you have a promoter that is methylated, it's going to be silent. If the promoter is not methylated, maybe silent or not through histone modifications. If the gene body is methylated, usually it corresponds to genes that are actively elongated or transcribed. So it depends on where the DNA methylation is taking place. It may have one role or another. Then we have multiple different histone modifications with different, let's say, non-redundant roles, and the combination of them defines particular states of the chromatin. We have the chromatin accessibility, where basically regulatory proteins are binding, and then we have the 3D interactions. And as I said, it's the combined action of all the different epigenetic players what provides the genome with function. Now, in this particular talk, I will focus, although let's say in the lab we've been focusing on all these different layers in the context of CLL, normal visual differentiation, and other lymphoid tumors. In this presentation, I will focus on DNA methylation. And DNA methylation, as I mentioned already in this presentation, has been related to gene regulation. If you read any review, or most of the great majority of the papers, they talk about DNA methylation as a major silencer, gene silencer. However, for this talk, and this is something that we have learned from the data, it seems that DNA methylation is able to track and to capture the memory of the cells from the very beginning until the moment that the leukemia is diagnosed. And this is something I will gradually explain to you through the analysis of DNA methylation from 3 different perspectives. So the first perspective is something we found back in almost 15 years ago, in which this is a work we did together with Marta Kulis, who was the first PhD student in the lab. So we analyzed a large series of CLLs with good clinical data, with the mutations as part of the ICGC Spanish consortium led at that time by Elias Campo and Carlos López Otín. So we had access to this incredible series, biologically and clinically very well characterized. And we did the DNA methylation. And what we found, something that really jumped into our eyes, is that there were two clear groups of CLLs with a different DNA methylome, the IEHB unmutated and the IEHB mutated, that actually contained traces, or where the differences between the two subtypes were related actually not to tumor suppressor genes or oncogenes, but to differences into the cell of origin, because the IEHB unmutated resemble more naive cells that have not gone through the germinal center, whereas the IEHB mutated CLLs resemble more like memory B cells that, of course, have experienced the germinal center reaction, and that's why they are mutated. Now, there was a third group in which we found that in part was resembling the memory-like and in part was resembling the germinal center inexperience, what we call this the naive-like. And this group, we call it intermediate because we didn't know which kind of cell of origin it could be. But the interesting thing, this intermediate epigenetics actually led to the identification of a third group of CLL in between the clear IEHB mutated or a clear unmutated, with this intermediate clinical behavior. And now, today, we know that the clinical behavior of this group actually can be explained by the presence of a particular immunogenetic mutation, the mutation in the R110 position of the Ig lambda 321, because when it is mutated, the clinical behavior is worse and will behave similar to the IEHB unmutated or the naive-like, and when it is not mutated, the intermediate group will behave actually quite well. So in terms of clinical behavior, there seems to be mostly two groups defined by epigenetics and immunogenetics. From the cell of origin perspective, the intermediate group we believe today is a group of cells whose cell of origin has gone through few rounds of germinal center maturations, few cycles between dark zone and light zone, and this cell has been selected to leave the germinal center reaction early, and that's why it has lower somatic hypermutation and a partial epigenetic programming that also occurs in the germinal center, whereas the memory-like have a high germinal center imprint and a higher DNA methylation and somatic hypermutation signature. Now, the second aspect, so if the first aspect seems to reflect kind of cell of origin, which is the maturation state of these groups of CLL, the three groups. The second aspect, that is a study that was conducted in the lab by a PhD at that time and now postdoc Martín-Subero, because there were, at that time, we saw that many of the changes in the DNA methylome were not related at all with cell of origin, were not related at all with gene expression, and we're like, what can that be? So we found there were some papers at that time that were saying, okay, DNA methylation can also act as a mitotic clock. The more the cells divide, the more they accumulate DNA methylation changes in the light replicating region. So we developed a kind of score that goes from 0 to 1. I will not go into the details, but it is a score that takes into consideration normal visual differentiation, takes into consideration the different chromatin states to select really heterochromatic regions and polycomb repressed regions. We select also regions that change during normal visual maturation, because if this proliferative history, we know a plasma cell has a very high proliferative history because it has gone through the germinal center and through past proliferative states, so it should carry that imprint of the past proliferation. And basically the score goes from 0 for stem cells to 1 to cells that have proliferated a lot in the past, because the proliferative history is cumulative. And I would like to differentiate between proliferative history and proliferative index, so Ki-67-like things. Ki-67, or proliferation index, is the proliferation of the cells you have when you take that particular biopsy or sample. Here, it is not that. It is the accumulation of how much the cells have divided in the past. That's why plasma cells, normal plasma cells in the bone marrow that are terminally differentiated and don't divide, they have the highest proliferative history, although they have a very low or 0 proliferative index. So that's important to differentiate, because with this we capture the history and not the present moment. So we applied this to multiple basal tumors, like 2000 in this paper, but I will show you here how incredibly powerful this is in terms to predict when the CLL patients will require the first treatment. So here you see the three colors, the green, the blue, and the red are the cells of origin I just mentioned in the previous slide. And the different shades reflect low and high proliferative history. And then you see how powerful the methodology, these two variables are to categorize patients into the likelihood of becoming, of progressing and requiring treatment. And the interesting thing here is not only, I mean, for the sake of that Kaplan-Meier curve, we separated this into groups, low and high proliferative history. However, in reality, this EPIGMET score is a quantitative trait that allows us to estimate when the patient will require treatment with quite a good accuracy. So if you put this variable as quantitative variable into a multivariate model, you see it is highly significant. You see the cell of origin with the three groups is also highly significant. And the number of genetic alterations, which is a kind of measure of genomic complexity, is also highly significant. But if you put together this one, together with this one, let's say two variables that are related to epigenetic memory, not necessarily are the cause of the progression, but more like the consequence. If the cells have grown a lot in the past, it will be captured in these two variables, and it will be a good predictor when the best predictor, these two together, of the time to first treatment. So, and this is a study we did together with Kathy Wu and Gadi Getz, in which we put together roughly 1,000 CLL cases in the CLL map. And then you can see here, this is a PCA, or principal component analysis, in which each dot is one sample. And if two dots are close to each other, it means that their DNA methylome is similar. And if they are far away, it means it is different. So this is done with two different technologies. One is based on arrays on the left. The other one is based on sequencing, NGS, on the right. And then you can see that the main sources of variability in the first two components are related to the two variables I just mentioned. The first one is cell of origin. You see the three clouds in both groups. But then you see a gradient from low here on the left to high on the right, which is basically the EPIGMET, or the proliferative history. So these two variables that are actually not related to gene expression are the two major sources of variability in DNA methylation, and both of them are independent prognostic variables in very strong prognostic variables in a multivariate tox regression model. Now, when we finished these papers, we thought, okay, we've learned all that we could about DNA methylation. So we were already focusing our attention to single cell technologies, ATAC-seq, histone modifications. But then, thanks to a common collaborator, Yotze Chibon, is a specialist in lymphoma biology and genetics at that time at Barts Institute in London. We were together in a consortium, and he said, well, there is this guy called Trevor Graham that just published something quite interesting on a particular type of DNA methylation, a new form of DNA methylation that they called it fluctuating DNA methylation. And then you have all this series and all this knowledge and all the clinical data on this, all this leukemia. So why don't you guys work together to do something different? And at this moment of my presentation, I would like you to read this quote from a man, as a British physicist that actually got the Nobel Prize by the age of 25. He's quite an intelligent guy. And he said something that I find it quite inspiring, because he said, in science, the important thing is not so much to obtain new facts, which we all like. We all like the new technologies, right? But he said, that's fine. But perhaps more important is to think or to discover new ways of thinking about the same data. And this is exactly what we did in this study that I'm going to present to you now. We basically took the same data published in this study of the proliferative history, roughly 2,000 B-cell tumors, different subtypes, acute lymphoblastic leukemia, multiple cell lymphoma, CLL, DLBCL, multiple myeloma. And we thought about them in a different way, using this concept of fluctuating CPGs. So I'll start. Now I ask you to pay attention, because this is the crucial slide to understand the concept. There is a lot of maths, very complex mathematics behind this. I will not explain anything about it. Just mention that it's going to be it's a model that is based on advanced Bayesian inference, and this is more the specialty of our collaborators from London. But I will explain you the concept, the concept is quite simple to understand. Now, this is the heat map with the fluctuating CPGs. And if you see a heat map like this, because when you see a heat map, in reality, you want to see patterns. Okay, here they're the different types of acute lymphoblastic leukemias, the different types of the CLLs, of the mantle cells, etc. But here it looked like random noise. It seems that there is no clustering based on subtype. It's kind of random clustering. But there is a pattern, and the pattern is that the normal cells are white. What is the color white in this heat map? The color white is 50% methylation. Now, the tumors have this speckled pattern. It looks like random noise of three states: methylated, 100% methylated, 50% methylated, or 0% methylated. And if you have a look at each different tumor, it's as if each different tumor, regardless of whether it's an ALL or myeloma or a CLL, has a different barcode, a different combination of methylated, partially methylated, unmethylated, and there is roughly 1,000 of these regions. So this is the identity of that particular tumor, right? So how can we understand, or how can we explain that in the normal cells, it is always 50% methylation, and in the tumor cells is 100, 50, or 0? Well, let me guide you through it. So these are normal cells. In let's say every column is one cell, up to millions of cells. In every row, it is one of these fluctuating CPGs. And if this is a polyclonal subpopulation, like in normal cells, they are polyclonal, they are not clonally related, every cell will have a different barcode. Every cell has a different identity. So if you do the mean of 1, 0.5, and 0, of course, the mean, if you do bulk methylation assay, both sequencing-based or microarray-based, you will see there is roughly 50% methylation for all the CPGs, and then the distribution will be monomodal. Now, if you take cell number 3, and with that cell, in our minds, we expand it clonally. And we imagine a very rapidly growing acute lymphoblastic leukemia. Of course, if that cell multiplies and grows exponentially, what will happen? Well, that barcode will multiply. And that barcode will be the only thing we have in the sample. If we have a sample that is containing all these acute lymphoblastic leukemia cells arising from one single cell, they all will have the same barcode. Therefore, cell number 1 will be methylated, cell number, or CPG1 will be methylated, CPG2 will be unmethylated, the third will be partially methylated, etc. So if we do the same bulk methylation assay, this CPG will be methylated completely, this one also, this will be partial methylated, this will be partially methylated, the others will be unmethylated. So at the end, we will have a W-shaped distribution for the unmethylated, the partial, and the completely methylated. If you find this in any type of cancer, in any type of sample that you don't know it's a cancer or not, imagine you do this and you find this W shape, this is a 100% sign of monoclonality. Now that's interesting and novel enough, I would say, but not the most interesting thing. The most interesting thing is that some of these CPGs They are called fluctuating CPGs. That means that they are relatively stable, but not so stable. So they can change over time. And as they can change over time, if one of the CPGs fluctuate, for instance, in this case, this CPG has fluctuated, has changed from the original one, all the daughter cells of that cell will carry that change. So there is room for evolution in the barcode. And when the barcode evolves over time, the distribution changes a little bit from 3 sharp peaks to like 3 broader peaks, and then sometimes they can move a little bit to the center. Now, from that distribution, we can fit a mathematical model that recapitulates that distribution. So basically, the model takes into consideration multiple parameters. So when the cancer the time since the most common recent ancestor, so when that cancer started to grow exponentially, the switching rates, the growth rates, so kind of models all these different parameters, and then makes thousands of different combinations. But that is one of those combinations that fits the observed data perfectly. So if we have then a model of how the evolution has been that fits perfectly the present moment of the sample, so the observed distribution, then we know all these evolutionary parameters, and we can estimate what was the patient age at which that particular cancer started growing, which has been the growth rate, the overall population size, the switching rates, and also whether there is even independent clones growing or subclonality. So I'll show you just a very brief example of all the data, and later on I will focus more on the CLL for the more clinical applications. So you see here, this is a scatter plot. It's not a PCA or UMAP. Here we represent the growth rate, and here we represent the overall population size. And look how the growth rate is of this group of samples as compared to these ones. Well, these are all the T and B acute lymphoblastic leukemias, which obviously they grow faster than the chronic B-cell or more mature B-cell tumors. So imagine that. So just think for a second that this plot comes out of this noise that is kind of goes through a very complex mathematical modeling, and then give us some specific values of the growth rate and et cetera, all these parameters. And then this is something that we can start validating. So this was the first slide we saw. Wow, this is really powerful. So at least it's capturing something that is quite expected out of the noise. And that's something that the eye cannot see. It's only through the bridge of the mathematics. The second thing we can do is we can take these fluctuating CPGs and make phylogenetic trees with them. So this is a case in which that was diagnosed 20 years, almost 20 years before the transformation to Richter. And we had samples. So in the hospital where I work, it's the hospital clinic and the IDIBAPS of Barcelona. They started decades ago this process of cryopreserving cells. So not only storing samples, but cryopreserving cells alive too, because they thought, okay, this is going to be essential. So we have an incredible sample bank of this sample. So we can do single cell, we can put the cells in culture, we can do many things with them. But in this particular case, there is 6 samples, cryopreserved cells from 6 samples, from the diagnosis to the moment of Richter, before and after multiple lines of treatment. So if we do the EvoFlux, and this is the barcode of the EvoFlux, here, these 2 samples of around time point 2 have identical barcode. Samples 5 and 6, you see the barcode is very similar, but has started to fluctuate. You see, this is different. Here there is some difference. Here there is some difference as well. Here as well. Now, if the Richter transformed cells would arise from the transformation of time point number 5, 0.4 years is roughly 3 months. The barcode in 3 months cannot change so much. In fact, in 3 months, there is no time for fluctuation. But the point here is that the barcode of the Richter sample obtained 3 months after time point number 5 is so different. And this is only compatible with one observation, and that it has been evolving over 30 years, that the Richter cells were present even 10 years before the initial diagnosis of CLL. And now you could think, well, this is something a bit like, difficult to believe, because we know this is a transformation of the other cells. Well, the question is it a transformation, or is something that has been there from the very beginning and somehow dormant or sleeping until those cells start growing as seeds that were sleeping and then started to grow much later, right? So this finding was quite interesting because the validation of this finding occurred a few years before, where Ferran Nadeo and Elias Campo, they published this study where they did whole genome sequencing, single cell sequencing, genomic sequencing, single cell transcriptomes of the very same samples, and identifying they determined the mutations of type 0.6, thinking that the mutations of type 0.6 were going to be an evolution of the mutations in type 0.5, but in reality, they found that the specific mutations of time point number 6 could be traced back to the moment of diagnosis in a very low, roughly 0.1% of the cells. So the mutations that actually define the Richter, or a fraction of them, were present from the very beginning. And also these cells seem to have a similar transcriptome. So it's interesting, right, that the Evoflux model, or the methylation model through this phylogenetic tree, was saying that these Richter cells are actually old, but they started to grow just 3 months after time point 5. And this is the validation that these cells existed at the moment of diagnosis. Of course, we don't know whether they existed 10 years before diagnosis because there is no sample, but that's what the model is saying. The last piece of information I would like to share is that imagine in the hospital, so there is 2 patients that arrive to the hematology service, and both have a CLL, and the CLLs both look kind of similar from clinical and biological perspectives. But imagine that one of them has been growing very, very slowly in the past to give rise to this leukemia at the present moment, and the other has been growing faster. Perhaps this was like growing 10 years, and this has been growing for 3 years. So when do you think that each of them will require treatment? Well, if you use all these evolutionary parameters to estimate the time to first treatment and the overall survival, you see that especially the growth rate was a massive predictor of when the patient will require treatment. And this is something you can see here, dividing to high and low growth rate in the IGHV unmutated and the mutated, and also how relevant this was in the context of P53 and IGHV mutations. Growth rate is an independent variable. P53 is not any more relevant, but these two are together quite powerful. So this is the article was published September of last year, received a very nice news and views, and the title of the news and views is quite revealing because it was saying like clues from the cancer's past foretell its future, and that clinged to my mind. Like I kind of read this somewhere, something similar, and then when I looked for it, it seems that it was said by Hippocrates a long time ago, who said, like the father of medicine, he said, in disease you have to declare the past. In that case, he was referring to talking with the patients, with anamnesis, right? But in this particular case, with these methodologies, we kind of force the cancer cells, to tell us which is their past, to be able to diagnose the present and to be able to foretell the future. So with this, I would like to finish saying that the DNA methylome is actually able to narrate the secret history of cancer cells and in CLL cells for this particular talk, although the principles are you can extrapolate the principles to other cancers, because they carry the imprints of the maturation state of the cell of origin and the type of cell. They can capture the overall proliferative history, when it started growing, what has been the proliferative rate, the overall cancer population size. And actually, if you put all these things into a multivariate model, they seem also all to contribute independently to predict the prognosis of the patients. So with this, our plan now is to build an individualized epigenetic memory score that captures all these things together. And final slide, just to thank the people who have done the work. This is our annual retreat in Montserrat, like a few weeks ago. This is the entire program coordinated and led by Elias Campo, that has it's a hematopathologist, but has a very broad vision on molecular aspects and clinical aspects of the disease. So he has built a program so that different groups tackle the same disease from a different perspective, and we all work together, trying to better understand disease, to better improve the clinical management. And this is a list of our many collaborators in the field of CLL. Their collaboration is really precious, and I very much enjoy collaborative research as compared to competitive research. And these are our funding agencies that help us with these experiments are not cheap, so they help us with some money and they believe in what we do and they help us to do it. And with this, I would like to finish. I thank you so much for your attention, and I hope we'll have a nice discussion.
Thank you very much, Professor Martín-Subero, for this insightful and very exciting presentation. So we'll now open the floor to questions. So please use the Q&A section to submit yours, and we'll share them with our speaker. So please.
Yes, feel free to ask questions. We are very lucky to have Iñaki in front of us, so please feel free. Maybe I will have a very first naive question so that it can be helpful for other people maybe to contribute to the next exchange. So first, one naive question I would have is that I've seen your very soft surrealistic clock from Dali on your very first slide, and wondering on these epigenetic clocks field. So something I wonder, because again, I'm not into epigenetic much. So looking at there is many works done on the epigenetic clocks and aging, and assuming that they pick up your blood and they say you're young or you're older or younger than your real chronological age. And if we do that, what I understand is that we should pay attention on the type of cells actually you are checking. And so that's my first, because you say that these marks depend on the differentiation. And also the second question I would have is whether the epigenetic clocks from the normal cells are different than the epigenetic clocks in the cancer cells, and could we apply the same tools in this post compartment?
Yeah, that's really interesting. And also in our papers, we also have we calculate all the Horvath clocks, the Hanum clocks, all the clocks related to chronological age and lifestyle. What do they tell us? So initially it was quite skeptical, because if you just put the methylation levels of those clocks, you don't see anything. Basically the statistical models are done so well that even if you take naive B cells from a 30-year-old and an 80-year-old, you do a heat map, they look very similar. The older one will have a little bit of demethylation in some regions, a little bit, but they look quite similar. But you apply the model, and the model will tell you 4 years and 81. So the models are quite accurate. So this is really interesting. And why is that? So my understanding of it is that they reflect if this is done with, let's say, with differentiated cells. The differentiated cells, they don't live for that long, so they are not subjected to the effect of time, but yes, the stem cells. So what I believe is that this is reflecting an epigenetic drift in the stem cells, so that they kind of, when they repopulate all the time the blood, with all the new cells will carry these small imprints of the more aged cells, and this is seen in the differentiated cells of the blood. Now, some individuals have epigenetic acceleration, and others are deacceleration. So we're talking not yet about cancer. Eh? That's the second question. That's what we'll answer later. So depending on the lifestyle and how well you treat your stem cells, how much you make them divide to fix damage in the context of inflammation, etc. So some individuals will be 10 years older than what they are, depending on how they have lived, and there are factors that really accelerate that, whereas others maybe 10 years younger, right? So that's quite exciting thing about lifestyle, etc., and how this impacts the state of the host. This has not really been integrated so well with the other clocks and with cancer, but it's definitely coming. And that's something we are super interested in the lab to focus on that. The other thing is that if you do the epigenetic age of the different cells of the body, they will tell you something similar. You take a fibroblast, you take a peripheral blood sample or different types of blood, will tell you something similar. However, the cancer cells, you take an acute lymphoblastic leukemia. In our case, we found some acute lymphoblastic leukemias that they are 200 years old.
Okay.
Yeah. So through the massive proliferation, etc., it seems that the model is seeing that there is a huge age acceleration. But now we know that cancer is not only related to age acceleration. There are some cancers that, for whatever reason, they are deaccelerated. So some cancers may be younger. Very few cancers have the same age as the normal cells. That's very rare. Usually in cancer cells, something is really upside down in terms when you do the epigenetic clocks for aging. Yeah. But we find very frequently age acceleration in the context of B-cell tumors. And the acute lymphoblastic leukemias are very extreme. Very extreme. Yeah.
Thank you. So yeah, there is also fancy questions from the Q&A. So maybe starting with the first one. So hello, just to be sure, for cancer samples, you observe 100% of this W-shaped for CPG distribution, ? And even on low priority samples, ? We can imagine these samples have a Gaussian shape for the CPG distribution.
No, the thing, if I understand it well, is the difference between if you have a pure sample or you have a sample that is contaminated with other cells. Yeah. In our study, things look so good because cells have been sorted. So for instance, CLLs is not just the peripheral blood sample. It has gone through MAX with CD19, and there is a selection. So the purity of the tumor cells is about 95%. The same for mantle cell lymphoma. The myelomas were CD138 pure. The acute lymphoblastic leukemia is not, but they are like full of blast, the majority, so their purity is high. So if you have a sample in which you have, imagine, 40% neoplastic cells, monoclonal cells, and the others are cells of the microenvironment. Or imagine you have a paraffin section, or you have a biopsy where you cannot do sorting. Things get complicated.
Thank you.
The W shape of the tumor cells will be blurred through all the other all the barcodes, individual barcodes of the polyclonal cells. So if you want to do this, I would say at least a 60 or 70% purity is very important. And if you can, in terms of leukemic samples, sorting or doing some MAX will help a lot, because the patterns you observe and the mathematical model and all the scores will be more accurate, and then they will work. The clinical predictions will be better if the samples are pure. Yeah, this is one of the caveats of the method. Although I believe that, for instance, technologies such as, for instance, long read sequencing, where you can perhaps separate the reads that come from a tumor versus the reads that come from the normal cells, and from the reads that come from the tumor, do the calculations, probably you can do the in silico purification. And that's something we have not yet tested, but I think is something that should be possible.
Okay, this is a very important message relating to purity and to clinical samples. So a second question, what does the growth rate represent here in EvoFlux? Is it like the evolutionary history or evolutionary index?
The growth rate. So for the model, for the math model, so the growth rate is more the slope of the exponential growth. So because the model assumes that the growth is at least at the beginning, before perhaps there is the cancer is like everywhere and the tumor, there's more control by the immune system. It's more the initial exponential, the slope of the exponential growth. Let's say, I would say it's more like the intrinsic capacity of those cells initially to grow. And this is because this is, for instance, not necessarily this has to correlate with, if you take different time points, it's not the growth rate of the different time points, because in the leukemias, sometimes the monoclonal cells in a CLL, they don't change over time. So is the growth rate zero? No, because I mean, these millions and millions and millions of leukemic cells in the bloodstream, they came out of one cell. So they're given initially in the silence of the apparent health of somebody, this cancer started growing very fast. So the growth rate is in the math model is this initial slope of the cancer growth. So reflecting more like the intrinsic capacity of growing. Yeah.
Another question.
That's our understanding.
There is plenty of questions. Huh? So next question is that, so try to summarize it. So could you comment on the expected informativity of the CpG methylation in diseases where we know there is alteration of such regulators, such as in AML with DNMT3A, IDH, and Z2 genes mutation?
Yeah, that's a very nice question. So the question is, will the presence of mutations of genes that actually modulate the DNA methylome, like Z2, DNMT3A, IDH, etc., affect the fluctuation rate, affect the way the mitotic clock is being manifested? The short answer is I don't know. I can elaborate a little bit more. In our EPICMID paper, in the proliferative history, we correlated it with the levels of all DNA methyltransferases, at least the levels of expression levels, not with mutations, because in CLL these mutations are very uncommon. They almost not exist. And there was not a big association. So there was a case had more DNMT3A or DNMT1. They did not seem to have a higher EPICMID in the hypermethylation or hypomethylation. We didn't find that. We have not done this systematically in AMLs, although it would be quite interesting to see in a control setting like AMLs with these mutations, whether we find something different, or even to do some in vitro models in which we introduce a particular mutation and we see how things change. So that's something I can imagine there could be an impact, but from the data I've seen in AML in other contexts, does not seem to be a big impact. But I think more control experiments have to be done.
Thank you.
Thank you. Now a question regarding technology and input sample. So do you think the epigenetic feature that you capture the tumor could be captured in the plasma as cell-free DNA? And if so, which technique would you the most promising in your mind?
Circulating free DNA, if the circulating free DNA comes, a large fraction of it comes from the tumor, they should work, right? In fact, there is people doing DNA methylation assays from circulating free DNA and they work. So we have not yet estimated all these parameters with cfDNA, but but it's something we are starting like some collaborations and thinking about that this could be a really good idea for non-invasive. For lymphomas, this is could be quite relevant, but also our collaborators is thinking more in solid tumors. So I think in terms of technology, I think a technology that probably, as the amount is so small, I wonder, this kind of arrays, we can do it with, if the quality is good, we can do it with 5 nanograms, 20 nanograms. For the circulating-free DNA, I think it's going to be probably better to do a capture-based method because we know the regions quite well. So I can imagine a capture-based method or amplicon-based method to capture these regions and then do the calculations after that. Yeah.
Thank you. So maybe a quick question. So you lovely will construct these phylogenetic trees on the basis of DNA methylation data. And is there any match or alignment with the classical reconstruction based on somatic mutations?
100%.
Oh, okay.
So this was one of the questions of a reviewer. Can you do the phylogenetic trees with the mutations? And of course, from these samples, we had whole genome sequencing. So we could do this with the more standard methods. And the shape of the tree is so similar.
Okay.
It's so similar. Yeah, very much. Very much. Yeah. This seems to capture evolutionary information the same way as mutations do. However, perhaps the virtue of this is that it allows us to do a lot of inferences and validate those inferences. And it's quite cost effective because it's relatively cheap to do. Yeah.
Thank you.
Maybe a question regarding Richter syndrome and long-term evolutions. So Richter subclonal and indirect evolution over 30 years looks very similar to follicular lymphomas with the concept of cancer precursor cells, so CPC. So in follicular lymphomas, the translocation 1418 and CREB-BP are the first 2 steps involved in the commitment to follicular lymphoma development. In CLL, so what is the rate of genetic events occurring during CLL progression versus the rate of the past, so the epigenetic past? In other words, do you compile genetics to your prognostic score? So how do you integrate epigenetic score with the other biomarker?
So, there are some considerations here. One of them is that the epigenetic memory variables and the scores we calculate, some of them, if you do like an oncoprint, they correlate with some of the mutations. So some mutations seem to confer higher proliferative rate. Some specific groups, depending on the immunogenetics, are also related to that. So in a way, we believe that the epigenetics is powerful because it's not driving. It is more like a silent observer of the cell's behavior. So in a way, higher proliferative rate comes out of which drivers those cells have, combination of drivers and deregulated cell cycle, but at the same time, to which degree they escape from the immune system. So actually how the cells proliferate is a combination between your inner drive to grow, intrinsic mutations, and some epigenetic changes that can also be related to that, and also how much control by the immune system there is. So the epigenetics is powerful from the epigenetic memory perspective because it seems to capture how the cells behave, both by intrinsic and extrinsic features. Now, having said this, I don't think that this is going to replace anything, but more act as a complement to what it is done so far. So I think that in the near future, what we will see, thanks to these new technologies of nanopore, the 5-base sequencing, the 6-letter sequencing, that can do with this, just changing a little bit the library preparation and treating the DNA with some enzymes can give you the mutations, but also can give you all these things that we are talking about. So I do envision that the diagnostics will be like implementing mutations and then epigenetics through different technologies, but more that the new combined technologies will be implemented. And it's going to be quite important to define or to design very nice pipelines to be able to easily extract the genetic and epigenetic information. And with that information, build models that are based on whatever information that is more useful. What I can imagine that in some cases genetics may be more powerful. In other cases, the method we do could be more powerful than genetics. I don't know. Imagine that if from the genetics perspective, if you do whole genome sequencing, there is a fraction of CLS that are driverless. That they still we don't know which are the drivers. Don't have anything of the common things. So in those cases, our technology will work equally well. So I think it's a combination of both things, and start implementing this into real settings, clinical trials, routine settings, to start seeing which is the real benefit beyond academic publications. Yeah.
Thank you. Maybe last questions. So this is something also I was wondering. It's how actually these features are really results from heritability or plasticity, and there was a question about that in your EvoFlux you have plasticity within the monoclonal population. Is it linked to cell of origin differentiation, such as a GC to memory, or to a reversible cell state?
Yeah. So plasticity, I mean, here we can understand plasticity as the epigenetic plasticity of the CPGs to fluctuate. I don't think it's in terms of lineage of plasticity in terms of phenotype, because that's something I'm not aware of in at least in the case of CLL. Which is the cause of that plasticity? Why there is subclonality? For instance, if there is subclonality and the both subclones are growing equally well, we will not see it. But if one subclone is growing faster than the other, we'll be able to capture with our method. So which are the causes of the fluctuation? It's just time. It's not only time. We don't know whether the fluctuation rates will be different in tumors that arise from precursor cells or from more mature cells. From that perspective, the differentiation state from which the tumor cells arise may be related to the fluctuation rate. The proliferation itself may influence the fluctuation rate, because the fluctuation to be given, to the daughter cells will have to be, so the cell will have to be divided for that particular fluctuation to be selected. So proliferation is also another thing. So my answer will be that I believe that the state of differentiation of the cell of origin and how much the cells have proliferated, I think, could determine the fluctuation rate and as such the EvoFlux calculations.
Thank you. So it's 2 p.m., so it is time to close the session. But I really want to thank Iñaki for this fascinating and super clear presentation, and thank you for making us understand very complicated stuff. And I also want to thank all the participants who attended today. And you will receive a post-webinar survey, so please fill it accordingly. And the next episode of this season 6 is April 28th, and it will be on organoids. So also plan to register and attend to this next meeting. Thank you very much for watching and contributing to the session.